Okay, hi. So my name is Tom Bultreys. I work at the Department of Geology, and I'm going to talk about storing CO2 in underground rock formations. So we all know about global warming, and we all know that we have to limit our CO2 emission. But while we're transitioning away from fossil fuels, one of the things that could help with that would be to capture CO2 at large CO2 producing power plants, for example, and to store the CO2 somewhere rather than to emit it into the atmosphere. Now, of course, we're talking about huge volumes of CO2 that have to be stored, and there aren't so many places where we can put this. But one of the possibilities would be to put it where we actually found the carbon in the first place, namely geological reservoirs. So if you have a geological reservoir, like an oil reservoir, for example, it consists out of a porous rock layer bounded by impermeable rock layers, and the fluids in the reservoir can actually be found within the pores of the porous rock layer. So if we have a CO2 storage site, then usually the pores will be filled with water before we inject CO2, and we will have to push the water away. And so if you want to study how much CO2 we can store, how quickly we can inject it, and whether or not it will remain safely in place, we really need to study the microstructure of these rocks, and more specifically the pores of the rocks in which the fluids can actually be found. And that's exactly what I've been doing during my PhD. So I investigate the simultaneous flow of two fluids in rocks. And first I'll show you a little bit about how we probe the microstructure of these rocks. Then I'll show you how we can learn about the physics of the fluid injection by doing fast imaging experiments. And finally, I'll also show you how we can simulate this injection. So let's start with probing the microstructure. So like I just said, it's the microscopic rock structure that determines how CO2 and water move within the reservoir. And to look at the microstructure, we use a 3D microscopy technique called microcomputed tomography, or micro-CT. And with this technique, we can actually get quite high resolutions, but only if it takes samples that are a couple of millimeters large. Now, luckily, we can do this technique at our own university because we have a Center for X-ray Tomography at UGent, which develops and applies these micro-CT scanners and the software to work with the data. And the center, called UGCT, consists out of 3 groups from the Department of Physics, the Department of Geology, and also the Faculty of Bioengineering. So one thing that you really have to keep in mind when you're talking about the microstructure of rocks is that these are very complex. They're very heterogeneous. They contain features at multiple length scales, and the pores within these rocks are often connected in a very complicated way. So we really have to take that into account when we want to know how the fluids flow within these pores. So let's look at that. Now, if you look at this in a bit more detail at a high resolution, then you'll notice that when you're injecting CO2, it will actually go into the pores in fast jumps. It will move into a couple of pores very quickly in a couple of milliseconds. And the injection rate will not control this time, but it will control the frequency of the jumps. So if these jumps happen so quickly, then that might mean that we have viscous and inertial forces which come at play, which are classically not taken into account. And there might even be non-local forces working because fluid has to redistribute very quickly from one pore to another. And well, this behavior isn't very well understood. So one thing that could be very helpful here is to use our 3D microscopy technique to look at these fluids in a real rock rather than in a simple model like the one you can see over here, which is 2-dimensional. So if you try to do that, then one of the issues is that you need sufficient time resolution because you want to make an image before and after one individual jump. And at field scale relevant flow rates, that means you have to make an image quite quickly. Now, benchtop micro-CT scanners typically take at least 20 minutes to make an image. So you would usually have to go to a large particle accelerator, a synchrotron, to make images faster. Of course, the beam time that you get at these institutions is very limited. So what we've been doing at UGCT is developing scanners, benchtop scanners, which can make an image much faster in a matter of seconds or tens of seconds. And you can see it working over here. And on it, you can see a sample cell which contains a sandstone sample of about 6 millimeters diameter, in which we've injected fluids while making images. And these are the results of that. So you can see our model fluid going into the rock sample. It's actually lamp oil that we're injecting because that's a bit easier to work with than CO2. So it's a bit of model fluid there. At an injection rate that is relevant to the field scale. And we've been imaging this at 12 seconds per scan, which is the fastest pretty much that has been done with benchtop scanners for this kind of system. So what we can learn from the analysis of this experiment is that actually it seems like the viscous and inertial forces are not so important, and that the capillary forces are the ones that dominate this behavior. And that's actually good news for our models, because our models are capillary models. So let me explain how we simulate this. So one thing that is, again, really important here is that this intricate microstructure really complicates our simulations, because it's very computationally intensive to track all the interfaces between the fluids, between the two fluids. And because of that, we have to do some simplifications. So we start with our model from a micro CT scan, and we extract a simplified network of pores and connections between the pores. And that makes it much simpler to simulate this injection. Of course, in this network model, we try to capture the real geometry and connectivity as best as possible, and we can actually get quite realistic results with this. What I've been doing specifically during my PhD is extending this model to be able to include multiple pore scales. This is really important in some rock types, because sometimes you can't even simulate anything if you don't take into account the multiple scales. And of course, if you've done multi-scale modeling, then you know that it's very computationally intensive, so you have to be very careful about how you go about doing this. So what we've actually done is we extract a network, and we add extra links through which we can add information that we've gathered from other experiments at other resolutions. So that allows us to incorporate multiple scales into one model, with which we can do simulations of injections of fluid. So as an example, I brought a model that is extracted from a sample from Svalbard in the Norwegian Arctic. As you can see, it's a very beautiful place. And so this group here is under consideration as a CO2 reservoir to store CO2 produced by a power plant on a town on Svalbard. So the simulations on this rock sample really need to include the multiple scales in this rock. And through these simulations, we get some information on the pressures of the fluids over here, and also on how much the two fluids impede each other's flow, because the fingers of the two fluids that are intermingled kind of slow down each other. And that's the graph on the right over here. So to conclude, CO2 can be stored in underground rock formations if we understand how the fluids behave during the injection. That means that we need to understand the influence of the rock microstructure and of the fluid dynamics. And fast micro-CT scanning and micro-CT-based models are giving us insights into this rich behavior, which we can also apply in other fields like groundwater flow, for example. Thanks.
Questions? Yes, please.